Thank you very much, everyone, for coming. Today I'm presenting this study. I'm the one presenting, but of course, this is a collaborative study. This was done in collaboration with the Balo Group at the WIHI in Australia and the Lee Group at the UW University in the States. And today I'm basically going to present to you how we tried to mix together multiple layers of omics data with imaging data using UK Biobank. I'm pretty sure most of you already know what UK Biobank is. If you don't know, very quickly, it's a huge British repository of medical health record. And for almost 500,000 patients, we have genomics, we have metabolomics, we have their clinical data, we have clinical imaging on these patients. And what I particularly focus on, and we focus on at the Lowy Medical Research Institute, are the eyes. Now, part of the imaging that is available in UK Biobank is called OCT imaging. Now, we are very focused on the retina. The retina is the back of your eye. Now, when we look at the retina like this, and then we flip it upside down on the side, basically we can see the retina as different layers. What the OCT is looking at are these layers. We can basically slice the retina into sections, and for each of these sections, we can measure what you can see in this image here. And one measure that we can do when we have this OCT-B scan, we call it, we can measure retinal thickness. So basically, how thick is the retina at any given point, right? We can do this for all of our scan, and we basically have a 3D map of the retinal thickness and a shape of our retina. And why do we use retinal thickness is because it's a very bit dirty proxy of retinal health. We know that when the retina is suffering, the retina tends to go down. So what we truly wanted to do with this model, we kind of wanted to match lots of information that we can extract from UK Biobank with this information about retinal thickness that we can obtain on these images, and so we can have spatial retinal imaging information. So first thing we did, went to UK Biobank, extracted all of this information, their demographics, all of their genomics information, their metabolomics information. We have measured hematological and infection markers. Well, we extracted from UK Biobank, and all of their ICD-10 as well as ICD-9 codes, which we then combined into Phi codes. We then extracted every OCT scan available in the UK Biobank. This was available not for all the 500,000 people, unfortunately, but only on 10% on 56,000 patients, still quite a large sample size. And our collaborators at UW, what they did is that they developed a neural network method that basically managed to segment this imaging automatically, and they measure retinal thickness in each single position that we can measure with the OCT imaging. And I'm going to show you in a bit what that looks like. I'm not going to go into detail into it, but we also use this 2D's retinal imaging map also to create shapes of the retina. These were captured using functional principal components. Again, I won't have time to go into it, but that was also cool. And the main goal of the study, as I said before, was basically to cross them. So we tested the association of everything that is in that blue box with everything that is in that orange box to try to understand association. And this is basically what we did. We have multiple omics, and for each omics, we are performing different analysis, but they all converge in the middle to the retinal thickness, to the spatial retinal thickness measurements. What do I mean for spatial retinal thickness? What do we get out? So we have almost 55,000 patients with this information that we could extract. Now, retinal thickness was measured basically in this grid. So for each patient, we have something like this. We have a grid, and for each of these points, we have basically 32,000 pixels, reduced to a little bit less when we start cleaning the edge pixels. For pixels here, I'm going to use pixels, but for pixels, I mean position, right? It's just a position in this grid. After cleaning, we are left with a little bit less because some of the outside pixels were a bit unreliable on our measurement, especially on the neural network. So those are cleaned out. But basically, for each patient, we get something like this. So we have a 2D map. And what we are doing is that, take, for example, for the genetics, when I say we are performing a GWAS studies, we're not really performing one GWAS study. We are performing almost 30,000 GWAS studies because we are testing for association of all of 8 million SNPs that we can measure in the genome with each single one of these positions across all of these patients. So we're basically performing a GWAS for each one of these pixels across our man. But not only we're doing genomics, we're doing multi-omics. So we're testing multiple different layers that we could extract from UK Biobank. And for each one of them, we're basically doing an omic study to, again, each one of them of this position that we can see. Given that we have an enormous amount of results, we have basically condensed everything into a website. If you Google Retinomics, you will find it immediately. This is a website that you can just look up and you'll find basically all of our results. I'm going to have another slide about this one later, don't worry. Yeah, this is basically an interactive portal in which you can explore all of our results. First, covariance association, there are different factors we need to correct for. So for example, looking at the association between sex and retinal thickness. Now, for each of these covariates, here I'm presenting you two different plots. One of them is the effect size that, for example, sex, in this case, being a male compared to being a female, genetically defined, as or retinal thickness. The other one is the significance. This is a simple minus log 10 p-value. The bluer it is, the more significant it is. And as you can see, sex, height, refractive error, and age actually was associated with it in a non-linear fashion. That's why we have both age and age squared. They were all quite associated. And already what you can start seeing already from this very simplistic analysis is that there are different factors which impact retinal thickness, but not in the same way across the entire retina. They have a very specific spatial signal that if we tested it on what other papers have done previously, which was a general global measure of retinal thickness, usually using an average or even using very big quadrants. We have ways of dividing the retina into quadrants. You wouldn't exactly get this type of very complex shapes, which instead doing these spatial associations can reveal. So what happened when we perform, for example, GWAS? We look at the genomics data. Now, here I'm showing a Manhattan plot of the GWAS performed right on the dead center of the retina. We call it fovea. This is the point where usually like our eyes focuses the light. This is completely for our central vision. And when we do a GWAS at the very center, the GWAS looks like this. If you move a little bit and you look at another spot of the retina, you can tell that the GWAS already moves. The loci, some of them are the same, but some of them now are changed. Significance have changed. This means that different parts of the genome are potentially regulating the health or potentially even the morphogenesis of the retina in different ways. And again, if you move it again into another part of the retina, you will start seeing changes. So to summarize our genetic analysis, we found lots of different associations. As you can imagine, we had to correct for a very stringent Bonferroni threshold because we are doing so many associations. But basically, in total, what we found between the pixel-wise analysis and the functional principal components analysis were basically more than 300 loci. Sorry, almost 300 loci, of which 120 were novel. They were never found before, even by other analysis who tried to do the spatial analysis of this thing, but dividing them in quadrants. So truly having this finite scale really helped. Another interesting thing is that when we compare different ancestries, we could actually see differences across the ancestries, which was not super unexpected. Apart from looking how the Manhattan plot changes, now you can look at, if you take a SNP in a particular gene that you might be very interested to, now you can look not only whether that SNP is significant, but where is significant and how it is significant. So for example, this is a very, very well-known gene affecting this disease called age-related macular degeneration. This is a degenerative disease that comes with age, most common retinal disorder out there. And what we could see is that the risk allele of this SNP tended to thin the retina, but particularly it tended to thin it in what we call the parafoveal area, which is this area here, right here. And this is an effect that was completely new. It was never observed before. We know that this SNP is of risk for this disease, but we never observed that he had this particular spatial pattern. One thing that I should mention about this is that I forgot to mention at the beginning, we only have healthy people in this dataset. We excluded, and for healthy I mean healthy for their eyes, we excluded all people with self-reported or known or detected retinal disorders. So we try to do this analysis only on our best corrected healthy population for their eyes and their eyes morphology. For example, you can check other SNP, for example, for these SNPs associated with thyroid eye disease, and you can see this really weird effect like that divides the superior compared to the inferior quadrants. Oops. Or for example, this other SNP affecting optic disc morphology. And I really hope that these images that I'm already showing you here truly show the value of doing this spatial analysis, because unless we did something spatial, even if we divided the retina in the ways that I'm dividing it in here, I don't know whether you can see those lines. This is a very classic way of dividing the retina. You wouldn't really observe them. They are very specific, they're very non-linear, and they're not completely contained in these quadrants that we would use. Even when we checked for the general GWAS results across the entire retina, here I'm just simply plotting number of significant loci at every pixel or irritability at every pixel. We can see that both of these things changes as we move across the retina. We try to cluster retinal areas given what SNPs were associated with those pixels. So basically combine retinal areas which are homogeneous for their genetic determinants. And what we could see, we could already find clusters. This is this clustering in here. And you can find that we start seeing concentric clusters that basically expand. At the side, there is this theory that the retina, as it develops, it actually develops starting from the fovea, so from the very center, and then it expands around. And then we did the same exercise, but we flipped it. We tested, can we cluster SNPs given how they affect the retina, in which part and in which way. And we could find different SNP clusters that have those average effects that I'm showing in here. So we found, 10 clusters, basically, of different SNPs that then we could even group into what genes they are and what gene sets do they go into. So that was quite exciting. And then we asked, can we connect all of these SNPs into polygenic risk scores and genetic risk scores? And when we look at risk of diseases or other traits, what does it look like? For example, this is just the genetic risk of age-related macular degeneration. And here we can see that even people who do not have the disease yet, or they've not reported it yet, but they're just at risk because of their genetic risk, their retina starts to change already. And it changes in very particular places that we can plot here and we can actually observe. So they already start to have a thinner retina. Same thing, for example, for people with a high genetic risk of glaucoma. This is another very common eye disease. Or macular telangiectasia type 2, abbreviated as MACTEL. And again, it's very interesting to see this negative effect just in the parafovea with this stronger effect right here in effect size, less in significance. But this is the starting point of the disease. Usually MACTEL always starts from this area here and then expands. So it was quite interesting to see that there was a stronger effect size in that area, although not particularly significant compared to the other area. We can look at even like systemic diseases like type 2 diabetes and other ones, and we can still see that they have an effect. Now we looked at genetic risk. What about actual diseases? So we can actually measure this disease in this data set using ICD-10 codes. One of the things that we found is that when we average the effect that diseases have on the retina, you can see it in this volcano plot, most of the diseases actually ended up on the negative side. So on average, we can actually see that having a disorder or having been diagnosed with a disorder correlates with a thinner retina. Globally, here I'm just averaging the effect across the entire retina for every of these diseases, but in general, we can see that they all go down. We also did a overrepresentation analysis over all of the pixel of retina with the question of, we have so many analyses for each one of these pixels. What are the pixels that are mostly correlated with disease status? So that particular pixel has a huge burden of disease association. When you have a disease, that pixel is likely going to change. What are these pixels? So we perform an enrichment analysis across all pixels. And again, what we found was this very interesting ring corresponding to like the parafovea and bleeding basically out into a temporal perifoveal, when instead the nasal perifoveal did not seem to be as much impacted as this one. And we also observed the differences between the superior side compared to the inferior side, which was quite exciting to see. We did metabolomics. Again, this is the same analysis, lots of different metabolites. And again, I'm not going to go too much into details, but again, we found that basically all metabolites were associated. We perform again overrepresentation analysis. And again, we found that the parafovea was more influenced. but more as well this temporal side, all of this area here. And in particular, it was quite interesting to observe that this lower area here, again, similar to Mactel, was particularly impacted by metabolic changes. And we know that Mactel that always starts in that area is a complete metabolic disorder. We know now it's almost completely a systemic disorder. We know there is also a localized disorder, a localized defect going on in the retina, but we truly believe that there is mostly a systemic defect going on with the metabolism of this patient. How am I doing with time? 5 minutes left? All right, I'll be very quick. If you look at different metabolites, for example, what LDL cholesterol does, and again, we can see shapes, or for example, your amount of glucose. The higher your glucose, the thinner your retina, probably driven by a fact that glucose has on your metabolism and your general health. Omega-6 fatty acids, we know that omega fatty acids are quite good for your health, and in fact, more increases retinal thickness, which is quite nice. And then I just want to conclude showing the blood traits as well. UK Biobank makes available all these blood traits measuring different things. And for example, when you look at blood traits that, for example, might relate to immune response, for example, like leukocytes or neutrophils, we can see that even neutrophil counts has an effect on retinal thickness that is quite localized in those particular shapes. Or even when we combine them together and we create composite scores like the immune inflammation index, you can see that even a high immune inflammation index is connected with a thinner retina, and again, as a particular spatial pattern. So to summarize, we integrated multi-omics data with imaging data that was translated via AI to get the retinal thickness. We get extremely detailed spatial effect that can be achieved by mixing AI and multi-omics. Retinal health truly seems to be associated with many systemic factors that we wouldn't expect. We don't expect everything to affect our eye. However, there is this new wave of publication now that are calling the retina the window to your body. We can actually see a lot of the things happening in our retina, and we can even predict the risk of stroke or even risk of neurodegenerative disorder in the brain by only looking at the retina. So this study confirms this new trend of findings. The retina is quite heterogeneous, especially, and the paraphobia we found to be particularly sensitive to changes, which is that ring that I keep talking about. And with that one, thank you so much. Happy to take some questions. And again, if you're interested, just Google Retinomics and you're going to be able to find us.
Thank you very much. Thank you very much, Roberto. There was a couple of questions.
Of course.
No. I will continue because I'm still in the neurodegenerative.
Okay. Hi. I think it's interesting, and I guess I'm just wondering, like, if I understand correctly, I think you're using each pixel to do the GWAS to associate with like different SNPs, for example.
Yes.
And I guess and I know that after that you do some clustering to see like the patterns. But I'm just wondering, have you considered like maybe cluster like different patterns of like the images or like the different region, like you mentioned first, and then do GWAS based on region to SNPs? Because for me, I guess it's a little hard to interpret how one pixel could be associated to one SNP. In contrast, like traditional GWAS, like when you do like when you try to associate one SNP to like phenotype, there's a kind of like underlying mechanism biologically that can lead to that, but just one pixel is a little hard. Or do you have any interpretation for that?
Yeah. I'm going to answer to your question. It's a very good question. Actually, something that I mentioned at the beginning, but I had to skip.
Okay.
This was our functional principal component analysis. This is basically this one. What summarize it very, very quickly, what you do is that you take the retinal thickness, these 2D maps across all patients, and then what you do is that you treat every pixel as a variable, and you compute a principal components. However, the functional part here comes by the fact that your algorithm now knows that these two variables, these two pixels, they are next to each other. So they have more weight to be pulled together by your PC rather than just looking at whatever explains most of the variability. So it's a mixture of things. When you do this functional principal component, what you obtain, what we have obtained, for example, are these 6 functional principal components, and what they measure are literally shapes.
I see.
We are literally measuring shapes. And we saw that when we do GWAS on every single pixel, we have a certain amount of SNPs, a lot of them that are significant. When we do functional principal components, we have still a lot. Some of them are different. We have found SNPs using these shapes that we couldn't found using the pixel and grid analysis. Yeah.
Okay, thank you.
Great talk.
Thank you.
My being in cancer, I think of retinoblastoma.
Oh yeah.
And so I was wondering if you look at not just the thickness, but even like heterogeneity of like cells on the surface, and then link that to like mutations, and then look at that in retinal. And if you've thought about that or have done that.
Again, a very nice question. Remember at the beginning, no, in this case, so quickly to answer to your question, no. But when you look at this image, basically what we measure is retinal thickness, right? Which is this very big proxy. However, can you see that the retina is divided in layers?
Yeah.
Each one of these layers is basically mostly composed by one cell type.
Okay.
Some of them by multiple one, another one is a part of a cell, right? We are about to embark on our next project, which is this project times 7. So we're going to do this same study now in each single layer.
Okay.
At that point, probably we're going to look at more genetic effect on particular cell types, because they can be divided by each layer, which might relate more to retinal health related to cancer, potentially by genes.
Great. Thank you.
You're very welcome.